Okay, now we're on to chapter 4. This chapter will look at tissues. Just to recap, you've looked at chapter 1, which was an introduction to the terminology related to anatomy and physiology. Then you looked at chapter 2, which went over chemistry and biochemistry. Then finally chapter 3, which went over cells, including topics like mitosis and DNA replication, transcription, and translation. So we're finally combining or moving up that organizational hierarchy and talking about tissues. Well, what are tissues? To remind ourselves, back in chapter 1, when we looked at that hierarchy of organization, we talked about tissues being a group of cells similar in structure and function, and oftentimes they look similar as well because of that similarity in structure. And we're going to see that their structure dictates their function, so we're bringing back that complementarity of structure and function tenet. The 4 major types of tissues are epithelial tissue. You may call this epithelium as well. So epithelial and epithelium are the same thing. There's connective tissue, muscle tissue, and nerve tissue. We're going to pay the most attention probably to epithelial and connective. They're the broadest of the 4 major types. If you need a general term for what does epithelial tissue do, well, it covers. It covers and it lines cavities, or it covers the outside of the human body. Connective tissue is by far the biggest of these categories, and connective tissue is there for support. Oftentimes, wherever you find epithelia, you'll also find connective tissue under that for support. Then we have 3 types of muscle tissue, which we'll look at. They all can contract, and because muscles, like the skeletal muscles, for example, can pull on levers like bones, they allow for movement. The cells themselves can also shorten or twist, and that is movement or contraction. And the nervous system is one of the systems that is known for control of the human body. The other would be the endocrine system, but at the very base level, cells that or tissues that are made up of these nervous cells are going to allow for a certain type of control. That's usually an electrical control via the use also of secretions called neurotransmitters. So we'll expound, we'll get bigger, we'll look at larger components of these 4 major types. But before you go any further, I would write these on a card. I would get used to them being the 4 major types of tissues. I'd get used to being able to list them off the top of your head. You should know the 4 major tissue types. And of course, we know that body cells are specialized to perform specialized functions, and they're going to help the body maintain homeostasis. And this just goes back again to that complementarity of structure and function. That tenet is going to underlie this chapter. We'll see not only that these cells look different, that they behave differently, they organize themselves differently, and they all have different functions to a certain degree. But we're starting really simple, 4 major tissue types: epithelia or epithelium, connective, muscle, and nervous tissue. So first let's talk about histology. What is histology? Very simply, it's the study of tissues. So histology is the study of tissues, just like cytology is the study of cells. So this is made up of the prefix histo and the suffix ology, and that suffix ology always means the study of. It definitely complements gross anatomy because we're talking about tissues, and you need at least 2 tissues. Usually there's all 4 types of tissues, but at least 2 in order to build an organ. And gross anatomy is looking at the organs in the human body, looking at how they're arranged. Maybe they're in the thoracic cavity, or maybe we're looking at a lower limb. Maybe we're looking at just a small compartment, but with gross anatomy, we can see things with our naked eye. And this is definitely going to provide the basis for understanding for the physiology of organs. What that means is that we know that most organs contain all 4 tissue types. So if we know how those tissues behave, we have an idea to how those organs are going to behave or how they're going to function. And again, this is going right back to that complementarity of structure and function, that what something looks like dictates how it works or how it's able to work because of what it's made up of. And there it is, finally, complementarity. We're going to see this really throughout the rest of this course. It's already underlined a lot of our chapters, but we got a little sidetracked with chemistry and then looking specifically at cells. But that complementarity of structure and function, that characteristic tissue makeup of organs, the arrangement of those tissues in organs, that structure is definitely going to protect function. So here's a little bit more on the tissue types. So we had our 4 basic types, we listed them, and that was a really simple list. So let's expand just a little further. Let's add just a little bit more information. So first, nervous tissue, our first word we were given for it was control. Well, how can it control things? It controls the human body via internal communication, usually via electrical signal and secretion of chemicals like neurotransmitters. What types of things are made up of nervous tissue, or what types of organs are made up of nervous tissue? The brain, the spinal cord, both of which sit in that dorsal body cavity, and then your nerves, sometimes called the peripheral nerves, that travel throughout your entire body. What about muscle tissue? We know it allows for movement. The reason it allows for movement is because of contraction. There's actually tiny filaments inside muscles that slide past each other and cause that contraction, which causes movement. There's different types of muscle. There's skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is attached to bones. Cardiac muscle is only found in the heart, whereas the walls of hollow organs, like your blood vessel walls, the walls of your stomach, or things like your small intestine, all of those organs, they have smooth muscle in order to allow them to move or contract. And there's different types of movements there, like segmentation and peristalsis, that would be able to describe the contraction of smooth muscle. Then you have epithelial tissue, or we could say epithelium. We know it's a covering, but it's both an inside and an outside covering. It forms boundaries. It makes compartments. It's protective if it's thick. It's able to contain cells that secrete. There's different shapes of epithelial cells, and the common functions you see there: protects, secretes, absorbs, filter, or excretes. Secretes, protects, or absorbs are really common for epithelial tissue. Of course, we can find epithelial tissue on our skin surface. That very top layer of our skin, of our integument, is made up of epithelial cells. Under that would be connective tissue, and then some fat, which is also connective tissue. But we can't just find skin or epithelial cells on the outside of the body. We also find it lining the inside of the stomach. It lines the inside of blood vessels, inside of the small intestine. So you can see that a lot of our hollow organs have both layers of muscle and epithelial tissue, which makes sense because they're organs. They're going to have at least 2 types of tissue making them up, usually all 4. We can also talk about connective tissue, and connective tissue's name is very apt. It connects things. It supports, it protects, it binds, it holds stuff together. It's by far the most diverse because it includes things like bone, blood, but also fat or tendons. So there's going to be tons of different types of connective tissue. It's the biggest category of tissue types. And maybe you're thinking, oh, I thought that was epithelial tissue. If I were to ask what is the biggest organ of the human body, that would be the skin. And that's where the confusion is coming, because you think skin, epithelial tissue, so of course epithelial tissue is the biggest tissue type. That's not the case, because most of your skin is actually connective tissue. Only that top layer is epithelial tissue. So the biggest organ in the human body is the integument, which you may call your skin, but only really the surface of your skin is made up of epithelial tissue. But the biggest category and the most abundant tissue type in the human body is connective tissue. So I'll say it again. The most abundant tissue type in the human body is connective tissue. So let's look at epithelial tissue and then contrast that with connective tissue. Again, epithelium, same thing. I sometimes see a lot of confusion there. There's different ways to say it. We could say epithelial tissue, or we're looking at epithelium. And epithelial tissue is usually going to form a sheet. We have cells that fit closely together. They usually fit in a nice little line. We find tight junctions commonly between epithelial cells. A lot of times these epithelial tissue sheets are impermeable because of those tight junctions. And there's 2 main types of epithelial tissue depending on their location. So you have the type of tissue that's epithelial and covers, or lines. That really forms a protective boundary or layer. And we see a lot of that in the cardiovascular system, the digestive system, and the respiratory system. And then you have a glandular epithelium, which makes up glands. But a lot of glands have these sheets of epithelial cells that are thicker than some of our covering and lining epithelium and are able to both form and secrete their products into gland ducts and then out of the body. These are just general functions for epithelial tissue, but you should recognize them if you had, say, a multiple choice question. You should be able to recognize all of these as a function that epithelial tissue can do. All right, protection. Real easy to remember because we have the integument. We have that top layer of our skin. It's actually made up of many layers of flat epithelial cells. So it's really protective because if something happens to that first layer of flat epithelial cells, you have plenty of more layers, and those are going to protect you. Absorption. We have cells in your digestive tract, mainly in your small intestine, that are able to absorb the things your body breaks down. Filtration. Your kidneys are really big filters, essentially, and they're able to perform this via a tube structure that's lined with epithelial cells. Excretion, also the kidneys. So the kidneys not only filter, they get rid of waste products. And that tubular structure that can filter can also get rid of substances, and that would be called excretion. We have secretion, and that's going to be our glands that are able to secrete their product through their cells. And sensory reception. So some of our sensory receptors, even though they communicate with neurons, are modified and made up of epithelial tissue. So let's just talk about the characteristics of epithelial tissue, how it exists, what it looks like. We mentioned that epithelial tissue likes to form sheets. So we actually can see here we have epithelial cells and they're lining up next to each other. And think of this three-dimensionally. So not only are they lining up next to each other, but they're also lining up in file. So think three-dimensionally. So we really do have a full sheet of cells. And whether they're cube-shaped or flat or maybe column-shaped, whether we have one layer or many layers, we're still forming a sheet. And what this gives epithelial tissue is a polarity. And if you remember something being polar back in chapter 2, it had two sides, a negative and a positive, slightly electronegative, slightly electropositive. This is kind of the same idea. We don't have any charges, but we still have two sides, two separate areas. We essentially have a top and a bottom, which is great terminology for structures like these. But we actually want to consider what you might call the top, just the free surface, the surface that isn't attached to connective tissue. So notice that in all four of these examples, that the epithelial cells are in pink, and then there is a dark purple layer under all the epithelial cells. This dark purple layer is connective tissue, and under all epithelial sheets, you would find a layer of connective tissue. Now, it doesn't matter which type of connective tissue right now, but just know that under all epithelial sheets, you would find connective tissue. And so the area where you don't find the connective tissue is considered the free region. In this example, it's the upper region, or you might want to call it the top region. But the free region is called the apical surface, whereas the non-free region, the region that is attached or lower or at the bottom, that's called the basal surface. So I'll point out the apical and the basal surface for all four of these examples. So this is apical, this is basal, apical, basal, apical, basal, and of course apical, basal. So all epithelial sheets have a polarity, meaning they have two separate sides that are slightly different, a free versus an attached surface. The free surface is called the apical surface, and that's where you might find things like microvilli or cilia. Whereas the basal surface, sometimes called the basal lamina, is where you're going to find connective tissue. Mainly where you're going to find things like glycoproteins, collagen fibers, that's usually adjacent to the basal surface, and again, those are going to be components of connective tissue. The basal surface connective tissue is not only giving support, because we know connective tissue is supportive, but it also acts as a selective filter. If something can't get past that connective tissue layer, it's never going to get to the epithelial cells from the bottom or from below that epithelial sheet. So just remember that epithelial tissue is polar because it has an apical and basal layer. Now I'm going to point out when we get to simple cuboidal tissue, because oftentimes this forms circles, that this idea of the apical surface is always up top and the basal surface is always at the bottom will not always be correct. It depends on the orientation of your epithelial sheet. Now, if you have a flat epithelial sheet, then top and bottom for apical and basal respectively works perfectly fine. But just be aware, that may not always work for you. So pay attention when I come back to polarity when we get to simple cuboidal tissue. Alright, here are some more characteristics of epithelial tissue. We know that we form sheets of epithelial tissue, so it should make sense that the cells are closely packed. There's some exceptions there for your glandular tissue, but for the most part we have tight junctions and even desmosomes in or between our epithelial cells in order to create a very tightly packed tissue. It's usually going to be able to filter or secrete or form a protective layer, so it makes sense that we want all of these cells very tightly packed. So let's look at this picture over here on the right. We can see again the apical free surface, the bound basal surface or attached basal surface, and then we have our sheet or layer of epithelial cells, or epithelium. In their lateral surfaces is where you would find the desmosomes and the tight junctions, because as types of cell junctions, tight junctions and desmosomes hold adjacent cells together. Tight junctions were our impermeable cell junctions. Desmosomes were our molecular zippers. If you need to review that, go back and check chapter 3. The other characteristic of epithelial tissue is the fact that all epithelial sheets are supported by connective tissue. And I mentioned the basal lamina. That's going to be the attached layer that's just below epithelial sheets. There's also generally a reticular lamina. And the basal plus the reticular lamina makes up what we call a basement membrane. And all epithelial sheets have a basement membrane. So I'm going to go over that again because that could be a very easy question you may see on a test. A question could say, what makes up the basement membrane? And then you would answer the basal lamina layer and the reticular lamina layer. And this is extracellular material that belongs to that underlying connective tissue. So these are just the two layers that are immediately adjacent to that underside or at that basal layer or basal surface for an epithelial sheet. Epithelial tissue also has no blood vessels. That's what avascular means. So if a tissue was vascular, that means it has blood vessels. But avascular means it has no blood vessels. It does have nerves. There are nerves, so it's innervated. Innervated means that a tissue has nerves going to it. And it has a really high rate of regeneration. Most of the time, not only can we replace the lost or damaged cells, the cells that replace the lost or damaged cells are exactly like the cells that went missing or were damaged. But let's talk about some problems here with the avascular nature of epithelial cells. They're avascular, there's no blood vessels that go to them, so how do they get nutrients? Because blood carries things like oxygen and glucose, things that cells need. And if epithelial tissue is avascular, aren't they going to die almost as soon as they are made because they can't sustain metabolism without those nutrients? Well, what happens is the connective tissue is vascular, and the connective tissue is so close to the epithelial cells that the epithelial cells are able to get the nutrients they need from the blood vessels in the connective tissue. They're able to get these nutrients from blood vessels that are in connective tissue, but not epithelial tissue, via a process called diffusion. So the way that epithelial tissue receives nutrients is via diffusion from blood vessels in connective tissue that underlies all epithelial sheets. Alright, so now that we have some of those characteristics covered, let's talk about how are we naming the epithelial sheets, because they do look different sometimes. Let's go back to that image we had when we were talking about the first couple of characteristics for epithelial tissue. We have single layers of epithelial tissue. We have some that are many layers. Some of our cells are cube-shaped, or flat, or column-shaped. So how do we name or classify these type of epithelia? Well, two ways. We name them based on the number of layers, and we name them based on the shape of their cells. So if we just have a single layer of epithelia, we call that type of layer simple. If we have two or more layers, we call that type of layering stratified. So you can see that this one, this next one, and here, so the top two and the bottom left, they are all simple types of epithelial tissue. Again, we're also going to name types of epithelial tissue based on their shape, and these cells in the top left are cube-shaped, so they're cuboidal. The cells in the top right are flat, so that's called squamous. The cells in the bottom left are column-shaped or rectangular, so they are columnar. And then the cells on the bottom right, you might want to say cuboidal. That might be your first hint, because at the bottom, if we look next to this basal layer, cube-shaped cells, definitely. But if you have stratified epithelial tissue, so if you have epithelial tissue that has two or more layers, which this right here does qualify, you are going to name the cell shaped based solely on the shape of the cells at the very top, at the apical surface. So if stratified, epithelial tissue is named according to the apical layer of cells. And here in the bottom right, the apical layer of cells are flat. So this is stratified epithelial tissue, and the cells are squamous. Alright, so let's look at some other examples. So this is simple versus stratified. This is simple flat cells. So not only would this be simple epithelium, this would be simple squamous epithelium. Here's another example of stratified tissue, two or more layers. And if we look at the very top, the cells are flat, so this would actually be stratified squamous epithelium. So here are cell shapes. Squamous, again, is flat, almost scale-like, kind of looks like a cell pancake. Then you have cuboidal, which is cube-shaped, and columnar, which is column-shaped. Usually for squamous and cuboidal, you're going to find the nuclei in the center of the cell, not necessarily, but generally. And in cuboidal, we see that the nucleus tends to be more spherical. In squamous, if we weren't looking at a side view, the nucleus would be more spherical or circular, but a side view would show us that the nucleus is oblong. And then in a columnar cell, the nucleus is oblong and much skinnier if we were to look at an apical surface view. So now that we've gone over the characteristics, the shapes, how we name types of epithelial tissue, let's look at different types of epithelial tissue. So for each of the following types of epithelial tissue, you do need to know a general description, like squamous cells are flat, and if I say it's simple squamous epithelium, it's a single layer of flat cells. That's an example of a description. Pay attention to its function and also the general locations. So let's start with simple squamous epithelium. This is a side view of simple squamous epithelial cells in the lungs. So in the lungs, you have really thin epithelial membranes that ensure that air exchange, or oxygen and CO2 exchange, if you want to be really correct, can occur. So this is actually a pictomicrograph of the epithelial tissue that you have in the air sacs of your lungs. It's really thin again because we want to ensure a thin layer to make sure gas exchange happens in time. So you can even see that within these thin layers we have dark spots. Those dark spots are nuclei of the simple squamous cells. And a lot of these simple squamous cells together make up a sheet, so that's epithelium. And a general description, single layer of flattened cells, very disc-shaped central nuclei. They're thin, kind of, again, like cell pancakes, so they don't have a whole lot of cytoplasm. Very simple type of epithelium. One layer, flat cells. Areas that you could find this type of tissue, of course the lungs, that's the example we're being given, but also in the kidneys. You can find it in the lining of the heart, the lining of blood vessels, lining of lymphatic vessels. It's very good as a single, thin, smooth lining. It's used a lot of times to reduce friction between two layers of tissue. So you can also find it in the ventral body cavity making up all of the serosae, all of those serous membranes. Because it's so thin, simple squamous epithelium functions really well in diffusion and filtration. Diffusion could also be absorption depending on the direction of the nutrient or gas flow. It's not really good as protection. There's only one single layer of it. So if that layer is disrupted, it's destroyed as an epithelial protective sheet. So not good for protection. But instead, it's really good for diffusion and filtration. And in some cases, it's able to secrete a lubricating fluid. A case would be in serosae. They're able to make or generate the fluid that fills serous cavities. Other words that we can use that still mean simple squamous epithelium are the terms endothelium and mesothelium. Now, if you use the term endothelium or mesothelium, you're not only saying what we're looking at is simple squamous epithelium, you're also giving a location. So these terms do mean simple squamous epithelium, but in a specific location. For example, if you use the term endothelium, you are saying you're talking about simple squamous epithelium either lining the inside of blood vessels or lymphatic vessels, nowhere else. If you say endothelium, you're talking about simple squamous epithelium in those two locations. Capillaries are a type of blood vessel, so still a lining of a blood vessel, but they're actually so small they consist only of a layer of endothelium. That also happens to be where a lot of gas exchange takes place. So it makes sense that it's a thin membrane because that would speed up gas exchange because the gases, or if we were looking at nutrients, don't have to travel through a really thick layer of epithelial tissue. In serosae, these endothelia would reduce friction, but endothelium cells that line blood vessels and lymphatic vessels are very flat and smooth. So for example, as blood flows from the heart to blood vessels, the flat and smooth layers are going to ensure that no friction or bumps or interruptions into blood flow or lymph fluid flow are introduced. Then you also have mesothelium. If you use the term mesothelium, you mean simple squamous epithelium that is a middle covering. So you're specifically talking about the simple squamous epithelium that makes up serous membranes in the ventral body cavity. And serous membranes in the ventral body cavity are going to surround the lungs, the heart, and in the abdominal pelvic cavity, the abdominal pelvic organs. Let's look at just the different types of epithelial tissue past simple squamous. So I said when we get to simple cuboidal, I'll show you a kind of modification or exception to the apical as the top and basal as the bottom rule. So if you have epithelial cells that are arranged in a ring, oftentimes you'd have multiple epithelial cells arranged in a ring. And then between your rings is the connective tissue, the darker pink. And that means that the attached layer for this simple cuboidal epithelium from the kidney is actually on the outside, not the bottom, because there's not really a top and bottom here. But on the outside of this ring, that would be the basal layer, because we can see the connective tissue touching it. In the inside, that free space right here is touching this part of the cell. So this layer of these epithelial cells are apical, because they're open to the free space. So again, top for apical, bottom for basal, that'll work most of the time. But remember that the apical surface is actually just the free surface, the surface not attached to anything, whereas the basal surface is the attached surface. So the region attached to something else, like the basement membrane made up of connective tissue. Here we can see simple cuboidal epithelium, Simple cuboidal epithelium is one layer of cube-shaped cells. You can normally tell how many layers in a histomicrograph by looking at the nuclei. So I see no more than one layer of nuclei as we move around. And you can find simple cuboidal epithelium in areas such as kidney tubules. It's great for secretion and absorption, which is things that occur in kidney tubules. But you can also find this type of epithelium in really small glands that contain ducts. Their ducts are oftentimes made up of simple cuboidal epithelium. The secretion and absorption are definitely going to be the main functions. Next, we're going to look at simple columnar epithelium. And simple columnar epithelium is going to be a single layer of column-shaped cells. We can even see that these elongated cells have their nuclei relatively lined up, and their nuclei tend to be closer to the basal layer at the bottom. So here we can see that this side of the cells is attached to connective tissue. So this is the basal layer. The free side is the apical layer. Here's another apical layer. Here's another basal layer. So again, top and bottom doesn't always work depending on what you're looking at. Again, this type of tissue is great at absorption and secretion. It can secrete things like mucus, enzymes, other substances. These cells are so large that they can contain these secretions until they need to excrete them into an environment. So simple squamous would never be able to contain or hold these secretions before they excrete them. You'd find simple columnar epithelium in certain areas of the digestive tract, such as the stomach all the way to the anal canal. Some of it's in the gallbladder, some of it's in ducts, some of it's going to be a ciliated variety, or it would have on its apical surface cilia, and it would be able to move things upwards, for example, towards the mouth if it was in the bronchi. It's even in some areas of the uterus where it also has the cilia. So general areas, you may notice a trend so far. After we hit simple cuboidal, absorption and secretion seem to be a common thread for the function of epithelial tissue. Because these cells are so great at secretion, we oftentimes find in between them, so in between the lateral borders of these cells, unicellular secreting glands called goblet cells. Goblet cells are generally found among simple columnar epithelium, and they are mucus secreting cells. So if you're asked to name, for example, you could see on a test, what is the unicellular gland that secretes mucus? Your answer would be goblet cells. Here is pseudostratified columnar epithelium. Sometimes it has a longer name, which is pseudostratified ciliated columnar epithelium. And actually in the photomicrograph, we're looking at the ciliated type of pseudostratified. So up here is the free surface, so this is the apical layer. Down here is the bottom or connected attached basal layer connected to the basement membrane, made up again of those two layers, basal lamina and reticular lamina. And from here all the way up to the top, that's the pseudostratified epithelial layer. Notice that if you were to look at the nuclei, there's some down here, there's some here, there's some up here, and it looks like this must be a stratified tissue. There's many layers of nuclei that suggest there's many layers of cells. And what you're also seeing are these kind of almost opaque, clear cells, and these are goblet cells. So both simple columnar and pseudostratified would contain those mucus-secreting goblet cells. Both simple columnar and pseudostratified could contain cilia. However, most commonly you would see cilia on the pseudostratified. And again, we're going to see the same functions so far. Since we hit simple cuboidal, absorption and secretion have been our main functions. Whether it's secretion of mucus or even propulsion of mucus by the ciliary action of the cilia that are on both simple columnar and pseudostratified. The location tends to be in the respiratory tract. So in the upper respiratory tract, like the trachea, or around your larynx. Trachea is going to be a big location, but you'd also find some of this in really large glands, or maybe even the sperm-carrying ducts in males. but the trachea, upper respiratory tract, is a good general function to know for, or location to know for pseudostratified columnar epithelium. Now, I mentioned that this tissue looks stratified. It looks like it has a lot of layers, but there's this prefix in front of stratified, pseudo, which means false. So that means this tissue is not really stratified. And if it's not stratified, if it's not 2 or more layers, the only other option is simple. So believe it or not, pseudostratified columnar epithelium is only 1 layer of epithelial cells, but the arrangement of these layers is where the confusion is coming from. So let's move away from the photomicrograph and look over here at kind of the clip art version of pseudostratified. And if we look at the basal layer, notice that all of the cells that make up this tissue touch the basal layer. But not all of the cells touch the apical layer. Some of them get squeezed off. Only some of these cells touch the basal layer and the apical layer. And this random arrangement of the cells, where some of them are squeezed and short, therefore their nuclei are down close to the basal layer. Some of them are elongated, therefore their nuclei are close to the apical layer. This arrangement makes this tissue look stratified, but it's not. So pseudostratified ciliated columnar epithelium is not really stratified. All cells touch the basal layer, just not the apical layer, and that gives it this falsely stratified appearance. If we were putting this tissue in simple or stratified as a category, we would have to say it's truly a simple tissue. It's just tricking you. So just be aware of that. Take a good look at the structure of pseudostratified ciliated columnar epithelium. Also practice spelling pseudostratified. Alright, next let's look at our first stratified type of epithelium, and this is stratified squamous epithelium. And I know it looks like stuff's crazy going down here at the bottom near the basal layer. The basal layer is actually wavy, so if we followed it, it went up, then down, up, down, up, down. So we have a wavy basal layer. The cells look a little thicker. We have many layers of nuclei, and that continues all the way up. And the reason we're not calling this columnar or cuboidal is the top layer of these cells are really thin. We can actually see the really kind of squished nucleus in one of these top layers of the stratified squamous epithelium. A description would be a thick membrane made of many cell layers, and the cells are going to be relatively flat. The cells at the basal layer are going to be much more cuboidal or larger, whereas the cells at the top layer are going to be a lot thinner. The cells at the top layer are actually really close to dying. That's why you see a lot less nuclei, so they're less metabolically or mitotically active, whereas the ones at the bottom are going to be generating new cells, and they're definitely performing more metabolism. We would find stratified squamous epithelium in a lot of locations, but its main function is protection. So with that in mind, think of where you would need some protection, such as the inside lining of your mouth. If you were to eat nachos and they were really sharp, it could kind of push on the epithelial layer inside your mouth, and if it was a thin layer making up that epithelial layer, it's not very protective. So it's more protective to have a thick membrane in areas where abrasion or stress could be put on a tissue. So it very much protects underlying connective tissue in areas that are subjected to abrasion. There's 2 different forms of stratified squamous epithelium. What you're seeing here is the non-keratinized type of stratified squamous epithelium. What we would see on your integument, on that top layer in your epidermis, which is another location for stratified squamous epithelium, is a really thick layer of pink. So if we were looking at a photomicrograph of keratinized stratified squamous epithelium, again, we see that thick layer of pink. We'd see a thick layer of keratin, which is just more protection before you even get to the cells. The non-keratinized version tends to be inside the body, whereas the keratinized version tends to be outside the body. Other areas that you'd be able to find stratified squamous epithelium is not only in the mouth or on your skin, but in the vagina and also lining the first part of your esophagus. So mouth, lining the first part of your esophagus before you get past your larynx. And then, of course, on your skin. So the epidermis. Alright, Then we won't talk a whole lot about stratified cuboidal. If we look at its name, a description would be 2 or more layers of cuboidal cells. It's not found a lot in the body. It's really rare, maybe some sweat and mammary glands, but usually they're in areas where there's some transition from one type of epithelial cell to another, and that transition produces this short area where you'd find stratified cuboidal. Same thing for stratified columnar, really limited distribution in the body, also found in those transition areas between 2 other types of epithelia. You find a little bit in ducts, in glands, some in the throat, again in those transition areas. And most of the locations you find stratified columnar are in those transition areas between one type of tissue to another. So stratified columnar and stratified cuboidal, really rare in the human body. A description of stratified columnar would be 2 or more layers of column-shaped cells. So that moves us on to transitional epithelium. Transitional epithelium gets its name because it can change shape, so it can transition. Pretty simple. It can resemble both stratified squamous epithelium and stratified cuboidal. However, its stratified cuboidal appearance is slightly different than normal stratified cuboidal. Stratified cuboidal would really be a little more square, a little more angular in shape, whereas when transitional epithelium is not stretched, its stratified appearance has a dome shape on its apical surface. So this dome shape to a lot of the cells on their apical surface, that's a giveaway that this is transitional epithelium. You would find transitional epithelium throughout the bladder, throughout the urethra, the ureters, really a lot of the urinary system. You do not, however, find it in the kidneys. The reason that transitional epithelium is found in the urinary system is because of its ability to stretch. So it can stretch, it can distend, and when transitional epithelium distends or stretches, these cells lose their dome shape and they flatten out. So they really kind of get pulled taut, and it looks a lot like stratified squamous all the way through. So for example, when there's urine flowing from the kidneys to the bladder, the transitional epithelium in the ureters will stretch. When it is not carrying urine, they go back to this dome shape. So the function really is to stretch readily and permit distension of urinary organs that contain urine. So the kidneys make the urine, but it's only considered urine once it leaves. So that's why we only find transitional epithelium in the ureters, which are the tubes that connect the kidneys to the bladder itself, and the urethra is the tube that allows urine to leave the body. Alright, and that leads us to glandular epithelium. So we had absorption, secretion, protective epithelia, and then we had glandular epithelium as our 2 types. So let's just take a quick look at glandular epithelium. Now, all glandular epithelium secretes something. It might be more aqueous or maybe a little more oily, but they secrete something. There's a production, there's an action that glandular epithelium are capable of. Now, we would classify glandular epithelium depending on the site of their product release. Where or how are they releasing their product? For example, there are endocrine glands and exocrine glands. So endo, exo, those are the prefixes. And endocrine glands are considered internally secreting. They're going to secrete their fluids, their product, not only into the interstitial fluid, but eventually to the blood. Endocrine glands secrete hormones, whereas exocrine glands are externally secreting. They'll excrete things like saliva and mucus and sweat, and they'll secrete their products into a duct. So exocrine glands have ducts, and endocrine glands do not. And you may be thinking, I think I know what a duct is, but what is it? Well, a duct is a tube, usually made up of some epithelial cells, that is going to lead the product to a specific location, like for sweat, the surface of your skin, or the surface of the open area of an organ, called the lumen. So depending on the relative number of cells forming the gland, whether it's a unicellular gland or a multicellular gland, it's going to be a more complicated or less complicated duct system. But only exocrine glands have ducts. Endocrine glands do not have ducts. So we even have this short kind of abbreviation here. Endocrine, ductless, they secrete hormones. Exocrine, with ducts, they secrete things that are externally excreted, mucus, sweat. Sebum, which is another name for oil, saliva, and then even digestive juices. Glands like the pancreas not only can secrete hormones, but they can also secrete digestive juices. So it's not strange for some organs to be really big glands that secrete both hormones and exocrine secretions. So let's look at a couple of modes of secretion. We're going to focus mainly on exocrine types of glands because those are the ones that tend to be made up of epithelial tissue. So we have multicellular exocrine glands and we have unicellular exocrine glands. But we're going to focus on the multicellular because unicellular is pretty much just the goblet cells. So goblet cells are a unicellular mucus-secreting exocrine gland. But the multicellular exocrine glands include the merocrine glands and the holocrine glands. The merocrine glands secrete their product by exocytosis. And the merocrine glands are going to have their epithelial cells be perfectly fine after the exocytosis. Whereas the holocrine glands, they're going to secrete their cells via cell rupture. So when the secretion goes to the duct, there's going to be secretion plus cell fragments. So when I think of merocrine glands, I remember that merocrine glands merely secrete. That's all they do, merely secrete. No cell products. Holocrine glands explode. The cells die. The ones secreting their product die if they are a holocrine gland. That way, the secretion includes dead cell fragments and product. And we see this actually with sebaceous glands, which produce sebum, which is another name for oil. And so they secrete oil and dead cell fragments because they're holocrine glands. So let's look at a picture. So here's our merocrine gland on the left, holocrine gland on the right. Notice that all of our secretory cells that are merocrine are secreting via exocytosis. The secretion gathers in a lumen of the gland and then is moved or squeezed into a duct of this exocrine gland. On the right, the top layer of holocrine cells are secreting their product by lysing. These cells basically explode, they rupture, so not only is secretion being released, but dead cell fragments are being released, and that's what is traveling to the duct. So merocrine glands merely secrete, holocrine glands rupture. With merocrine glands, you get just the secretion to the duct. In holocrine glands, you get secretion plus dead cell fragments to the duct. Alright, and that is it for epithelial tissue. We looked at the characteristics, the naming, so classification, different types of epithelial tissue, all the way from simple squamous to pseudostratified and transitional. And we looked at glandular tissue also made up of epithelial sheets. The difference between merocrine and holocrine is a good focus for that glandular tissue part or unit. Next, we're going to move on to the connective tissue, which is our most abundant and widely distributed tissue type. So make sure to watch that video as well.